Hello, I'm Hiromichi Hoshina, Senior Research Scientist at RIKEN in Japan. We recently found that the actin protein, which plays an important role in cell functions, changing its morphology by the irradiation of terahertz waves. We think that's quite interesting because terahertz radiation, which is used for next wireless technology, may affect human health. We have published some papers and would like to review in this video titled Terahertz Radiation Changes the Protein Structure in Cells. First, let me talk about the research background. With the recent development of terahertz technology, a variety of applications have become possible. Devices used in the millimeter wave region have been upgraded to higher frequencies, and the prices are going down. As a result, terahertz waves are being used in our daily lives. I would like to show two examples for terahertz application. The first is a radar. For example, the anti-collision radar used in the automobiles is using millimeter waves around 0.1 THz. Recently, such radar devices have been used in the smartphones as motion sensor like Google's Siri. The second is the wireless communication. This figure shows the frequencies used in the cell phones. The frequencies become higher in the later generation to send a large amount of information. In the 6G regulation, it is said that the frequencies will enter the terahertz frequency region. As terahertz light become more and more common in our daily lives, you may be wondering if it is safe for the human body. Unfortunately, on the internet, we cannot expect correct answers, and rumors are widespread. For example, some people believe that terahertz waves are good for your health, and they are selling bracelets made by quartz, germanium, and silicon. On the other hand, there is an opinion that terahertz waves are bad for health. This picture shows that 5G antenna was burned down in UK last year. People believe that millimeter wave communication is the cause of the coronavirus. Of course, there is no scientific evidence for these rumors. Okay, then let's start scientific talk. When terahertz light is irradiated to the biological samples, thermal and non-thermal effect are induced. Thermal effect is dynamical excitation of biomolecules and surrounding water molecules and heating the system. Many researches have been performed for the thermal effect of terahertz irradiation. This figure summarizes those results. The red area is the irradiation condition of high power and long duration, which cause cell death and tissue damage. The yellow area is a lower power and short duration. Nothing changes. The purple area is in between. In this boundary condition, we often observe the production of the heat shock protein in the cell, which is originated in the heating stress. In some terahertz irradiation experiment, increase of the heat shock protein has been reported. Most probably, that's due to the thermal effect. On the other hand, non-thermal effect is not clearly understood. Here, I will propose some possibility of the non-thermal effect. The first one is the molecular orientation. When terahertz electric field is interacting with a dipole or induced dipole moment of biomolecules, those molecules might align like this. However, higher electric field is necessary for this phenomenon. Orientation of large molecule in the condensed phase is quite difficult. Molecular orientation have been observed only for the small molecules in the gas phase. Second case is the excitation of the molecular vibration. In the terahertz frequency region, corrective vibration of the intra- and intermolecular structure is observed. For example, this movie is taken from our recent paper in which corrective vibration of macromolecules and its bound water is simulated. Those vibrations are often observed in the terahertz region. Therefore, terahertz irradiation excites those modes. As a result, structural and dynamical change of macromolecules might occur. The third case is the mechanical effect. For example, shock waves can be generated by the pulse irradiation. When intense energy is irradiated to the sample in short time, like picosecond, the instantaneous concentration of the energy might cause generation of shock waves in the media, and the shock waves mechanically changes the structure and dynamics of macromolecules. Those non-thermal effect of the terahertz irradiation is in our research interest because not many things are clearly understood. Okay, then let's start talking about our research. First, I'd like to talk about our sample molecule, which is actin protein. This picture shows the fluorescence image of actin in the HeLa cell. The red structure is the actin filament that compose the cytoskeleton, and the green area is the actin monomer in the cytoplasm. In the cells, Monomer actin polymerize to the actin filaments, and the actin filaments are depolymerized again to monomer. Actin filament plays an important role for cell functions such as cytokinesis, cell migration, and cell invasion. By observing structure of actin filament under terahertz irradiation, we can see how terahertz light changes the actin morphology and cell functions in vivo. The good point of actin protein is that we can perform in vitro experiment by using purified actin. As shown in here, cycle reaction of actin filamentation can be reproduced in the bulk sample. We can observe this reaction under fluorescence microscope. By irradiating terahertz light during this reaction, we can observe the change of the reaction rate in the simplified system. As a result, we can understand the mechanism how terahertz light changes the actin polymerization. For the terahertz irradiation experiment, we used two types of terahertz radiation sources, terahertz pulse source and terahertz continuous wave source. As a terahertz pulse source, we used terahertz free electron laser at Osaka University. For the continuous source, we used gyrotron at Fukui University and impact diode, which is commercially available. First, I would like to talk about the radiation experiment of pulse terahertz waves. This picture shows the terahertz free electron laser at Osaka University. This laser is very powerful, and the energy of the output macropulse is about 50 mJ. Each macropulse is composed by hundreds of micropulses. The micropulse energy is about 500 microjoule. We can tune the center frequency of the output from 3 to 6 terahertz, but in our experiments, we fix the frequency at about 4 terahertz. Using terahertz free electron laser, we studied about irradiation of terahertz pulses with the filamentation reaction of the purified actin. This is an experimental setup. The actin solution is placed on the film bottom dish, which is transparent to the terahertz wave. The output of free electron laser is irradiated from the bottom. Since the output of FEL is too powerful, the energy of the irradiated terahertz beam has to be reduced. To observe only the non-thermal effect, the terahertz beam was loosely focused and reduced by wire grids and terahertz attenuator. As a result, the irradiated micropulse energy density was about 80 microjoule per square centimeter. Also, we estimated the sample temperature by using this model. This figure shows the temperature change under pulse irradiation. When terahertz pulse comes, the sample temperature rises about large delta T, like this. This value can be evaluated by adiabatic model as 0.04 degree per pulse. After the terahertz pulse leaves, the sample temperature drops by thermal diffusion. The average temperature rises slowly as small delta T, which can be measured by thermocouples as 1.4 degree in 30 minutes. From this estimation, we concluded that the temperature change on the sample is within a few degrees. That is not too large to change the rate of actin polymerization reaction. We started polymerization reaction from actin monomer under terahertz pulse irradiation. After 30 minutes from starting reaction, we picked sample to a fluorescence microscope and observed the fluorescence image of actin filaments. These pictures show the fluorescence image after 30 minutes, with and without terahertz pulse irradiation of 80 microjoule per square centimeter. We can clearly see the number of actin filaments decreases in 30 minutes. We counted the number of actin filaments for many pictures and summarized in this graph. The number of actin filaments clearly decreased. It looks terahertz pulse irradiation inhibits actin polymerization reaction. However, something is strange. In this experiment, the thickness of the sample is about 1 mm, which is too thick because the absorbance of the water is about 1000 inverse centimeter at 4 terahertz. That means 99% of the irradiated energy is absorbed within 20 micrometer depth. Therefore, less than 1% of the sample can directly interact with terahertz waves. That means the observed fragmentation of actin filament is not due to the direct interaction with terahertz wave. As a candidate of this indirect interaction, we think that something like laser-induced shock wave may happen. This figure shows the shock wave generated by femtosecond laser pulse. After a laser pulse is irradiated to the sample, plasma is generated at the focal point, and that produces shock wave in nanosecond to microsecond order. We think terahertz pulse might induce something similar phenomena. To demonstrate terahertz pulse making shock waves, we performed direct observation of shock waves induced by terahertz pulses using shadow graph method. This is an experimental setup. The output of the terahertz free electron laser is loosely focused on the water in the quartz cell, and the probe beam is irradiated horizontally to the cell. The shadow image of the probe beam is observed by CCD camera with image intensifier. With this setup, we measure the shadow pattern by 10 nanosecond step. This movie is made by the shadow graph images. The terahertz beam comes from the upside, and the shock wave is generated at the surface, which makes a stripe pattern. In this video, that is transmitting downward. The stripe pattern comes from the hundreds of micropulses in the output. This figure shows the expansion of this video. The distance between stripe is about 55 micrometer. The interval of micropulses is about 37 nanoseconds. Therefore, the speed of shock wave is obtained as 1500 meter per second, which is close to the sound velocity in water. By this experiment, we demonstrated that terahertz-induced shock wave propagates more than a few millimeters. That is the first candidate which demolishes the actin filaments. Previously, in the studies of shock wave generation, pulse laser has been used. When femtosecond laser is focused on the water surface, it makes plasma by ablation, and the pressure of plasma makes shock waves. It requires higher power density like petawatt per square centimeter. On the other hand, terahertz wave can directly excite intermolecular motion of water molecules. As a result, terahertz-induced shock waves requires less than gigawatt per square centimeter. The advantage of terahertz-induced shock wave is the effective energy conversion, because the energy of terahertz pulses is completely absorbed by water. Also, there is no photo damage to the sample. We think that terahertz pulse irradiation might become a new method for shock wave generation. Okay, let's go back to the irradiation experiment. Next, we try to observe the radiation effect of terahertz-induced shock waves for the actin filaments in the living cells. This is an experimental setup. HeLa cells were grown on the cover glass and placed like this. Actin filaments are stained by silicon rhodamine actin and observed by fluorescence microscope. Terahertz pulse from FEL is irradiated from the bottom, which is completely absorbed by water, and only the generated shock waves can reach to the sample. Sample temperature is controlled and rise of the temperature is negligible. These are the fluorescence images of the actin filaments in HeLa cells without and with terahertz irradiation of 80, 160, and 250 microjoule per square centimeter. Without terahertz irradiation, image of the actin filaments in the HeLa cells does not change so much. On the other hand, as shown in the red arrows, actin filaments are fragmented after 30 minutes of irradiation. That can also shown in the high-resolution images, in which actin filaments are fragmented or disappeared. We concluded that terahertz pulse demolishes the actin filaments in the living cells, even if those are placed a few millimeter depths in water. This is the conclusion of the first part. We found that shock waves are generated when aqueous solution is irradiated with terahertz pulses, and the generated shock waves propagate more than a few millimeter depths in water, which is longer distance than the thickness of human skin. The generated shock waves demolished actin filament in the living cells. We have thought that the irradiation effect of terahertz wave is limited only on the human body, because it's completely absorbed by water. So we said terahertz wave is not so dangerous for tissues inside the body. However, we found that in the case of pulsed terahertz waves, the energy can be converted to shock waves and propagate inside the human body. That can affect the human health. In the next part of this video, I would like to talk about terahertz irradiation by continuous wave sources. We use two types of terahertz sources. One is gyrotron at Fukui University, which is powerful, and the output of gyrotron is 0.46 terahertz, and the power is about 0.6 watts per square centimeter. The second one is commercial impact diode source, which produces 0.28 terahertz with a power of 0.1 watt per square centimeter. The size of the impact diode source is very compact, so we can build tabletop system for terahertz irradiation experiment. Irradiation by the continuous wave sources is totally different from the pulse sources. First of all, we have no shock wave because the peak power is very low, and also temperature rise is negligibly small. We observed actin polymerization reaction under continuous wave terahertz irradiation. This figure shows the polymerization reaction of actin and fluorescence intensity. At first, nucleation starts and after elongation, actin filaments reaches to the equilibrium. First, we irradiated terahertz during elongation process. This graph shows the fluorescence intensity with and without terahertz irradiation. It clearly shows that the terahertz irradiation accelerates filamentation of actin, which is completely opposite result with the pulse terahertz irradiation. These pictures show the fluorescence images at the end of the reaction. Clearly, the number of actin filaments are increasing with terahertz irradiation. Next, we irradiated terahertz after the sample reaches to the equilibrium. In this case, the fluorescence intensity increases by the terahertz irradiation, which means that the equilibrium condition changes with the terahertz irradiation. Most probably, polymerization or dissociation rate of the reaction changes with the radiation. We concluded that terahertz irradiation accelerates the actin polymerization reaction. We also performed the irradiation experiment for living cells. This figure shows the experimental setup. We irradiated continuous wave terahertz from the bottom of the sample and observed HeLa cells stained by silicon rhodamine actin. These are the fluorescence images. Without terahertz irradiation, the number of actin filaments does not change so much. On the other hand, with the terahertz irradiation, the brightness increases in this area, suggesting that the number of actin filaments are increasing. To show this result more clearly, we counted the fluorescence intensity for more than 100 cells and summarized in this graph. This graph shows the statistics of the fluorescence intensity for individual cells at 0 and 30 and 60 minutes. Without terahertz, the fluorescence intensity does not change so much, but with terahertz, it increased with time. Continuous wave terahertz irradiation clearly increased the number of actin filaments in the living HeLa cells. At the right side of this figure, we plot the result of same experiment with just platinolide, which is a drug for increasing actin filaments. Terahertz irradiation works same as this drug. The increase of the actin filament was also observed by the weaker irradiation condition. This graph shows the fluorescence intensity of actin filament in the HeLa cells with the irradiation of impact diode. The intensity increased slightly but clearly higher than the control samples. Here again, we can conclude that continuous terahertz wave induces the actin polymerization reaction and increase the actin filament in the living cells. The increase of the actin filament can change the cell function. For example, actin plays an important role for cytokinesis. Figure A shows the cell structure during the cytokinesis. To divide mother cell into two daughter cells, this red ring, named contractile ring, is formed and shrink. Actin filament is the main component of this ring. These pictures show the fluorescence image of the HeLa cells under cytokinesis. The red arrow shows the contractile rings. Without terahertz, the cells complete cytokinesis within 30 minutes. But with terahertz, it does not finish. Figure B shows the percentage of cells arrested in the cytokinesis. It clearly shows that terahertz radiation increases the number of cells under cytokinesis for more than 30 minutes. Most probably, Induction of actin filaments disturbs the decomposition of contractile rings. This is the conclusion of the second part. We found that the radiation of continuous wave terahertz accelerates the actin polymerization reaction. This phenomena is opposite of the heating effect because the actin polymerization reaction decreases by heating. Terahertz irradiation increases the number of actin filaments in the living cells and also changes the function of the cells. The contractile ring during the cytokinesis does not disappear under terahertz irradiation. We clearly show the terahertz irradiation inhibits cytokinesis, which means terahertz radiation affects for human health. Okay, let me summarize our findings. We found two different non-thermal effects of terahertz irradiation. The pulse terahertz irradiation induces demolition of actin filaments in vivo and in vitro. These phenomena were explained that the actin filament was demolished by shock waves, and the terahertz-induced shock waves were observed. This result shows that the terahertz pulse affects the human body by demolishing actin filaments. On the other hand, when we irradiated continuous wave terahertz, actin filamentation was induced. Increase of the actin filaments are observed in vitro and in vivo. We also found that the cytokinesis of the cell was inhibited by the irradiation. The mechanism is now still under research. This result shows that the terahertz continuous wave affects the human body by inducing the actin filamentation. At the end of this video, I'd like to thank all collaborating researchers, especially Professor Harata at Tohoku University and Professor Ogawa at Kyoto University, and Dr. Tsubouchi at QST, and Dr. Yamazaki at Riken. He is currently working at NICT in Japan. And also I'd like to thank these grants. Thank you very much.
Before we go ahead, don't forget to click the subscribe button on my channel.